as I said, you should be able to see an appetizing meal on your screen. The application of genetic engineering techniques to agriculture and food production has been a hot topic for almost 30 years now. All the public have yet to be persuaded that the potential benefits outweigh the perceived risks. Recently, however, a new and more pressing factor has entered into that equation. Hang on. Right, and this is it. The deadly interplay between population growth and climate change. The UN estimates that by 2050, there will be 9.7 billion of us on this planet. That's over 20% more than the 7.9 billion that there are today. And all of those extra mouths will need feeding. However, if we don't address climate change now, not just the relatively modest proposals agreed at COP26, but also all the other contentious ones that were kicked down the road, then crop yields are set to fall globally by about 25% over the same period, all due to climate change. The consequences, needless to say, would be catastrophic. But if you're thinking that climate change is entirely down to our reluctance to abandon fossil fuels for energy and transportation, rather than anything that farmers and the food industry are doing, this next chart should make you think again. A quarter of all greenhouse gas emissions arise directly or indirectly from the food industry. More than half of these emissions are related to animal products, of which half again are attributable to cattle and sheep. All of which means that, because they're part of the problem, the food industry will have to be part of the solution as well. But more about that in a moment. First, take a look at this. This chart compares, for a range of different foodstuffs, how much land is required to produce just 1 kilogram of the product. And some of these numbers are truly staggering. Way out in front are beef and lamb, both of which swallow up more than 300 square meters of pasture land in order to produce just 1 kilogram of meat. Pigs and poultry are more economical, requiring 10 to 20 square meters per kilogram, but that's still a great deal when you compare it with grains, vegetables and fruit, all down here at the bottom, requiring less than 5 square meters, often a lot less, to produce a kilogram of food. The message is clear, but if you still need further convincing, take a look at this next chart. This is the corresponding data for water usage, expressed as litres per kilogram of product. Calculations here are more complicated because of the various ways that water is used, but the key message is the same. Meat products in the top half of the chart use far more fresh water than grains, vegetables and fruit, which, with the exception of rice and also nuts, are all concentrated at the bottom half. And so, incidentally, is wine, so you won't need to give that up. So to sum up, in a warming world with a growing population, but with a limited amount of cultivatable land and increasingly uncertain rainfall patterns, we have to significantly reduce our dependence on livestock farming and adapt to an increasingly plant-based diet. Easy to say, of course, but for all sorts of reasons, economic, social and political, much harder to achieve, and certainly not in the limited time we have left to make enough of a difference to avert the impending global food crisis. So we do need to consider other options as well, and these are just some of them. As we've seen, our behaviour is simply going to have to change, not just by cutting back on meat, but also ensuring that our increasingly plant-based diet is primarily sourced from produce grown here in the UK. By importing food, we contribute to global warming because of the hefty environmental impact of international shipping. And all of us can help by not wasting food. Did you know that more than a third of all food produced globally is never actually eaten? In the UK alone, we throw away nearly 10 million tonnes of food waste every year. Much of it still perfectly edible. These are things we can all do, but just as important are things which only the food and farming industries themselves can do to ensure that our agriculture is sustainable for the long term. Pesticides and fertilizers, because of the way they're manufactured, have significant carbon footprints, and adverse ecological effects, we really need to find ways to cut back on their use. Fertile soil is our greatest agricultural asset, but it's easily damaged. We need to take care of it by moving away from monocultures, huge fields planted with the same crop every year, and start to adopt the principles of regenerative agriculture, which focus on protecting soil structure, fertility, and resilience. And last, but by no means least, as custodians of the planet, we're responsible for preserving its biodiversity. You've only got to look at the steep decline in the populations of pollinators, particularly bees, here and around the world to appreciate how damaging this could be. And finally, we need to be prepared to embrace innovations, whether it be alternative sources of food like algae or insects, or as here, technological advances. So if you really can't face giving up meat, there are now synthetic laboratory-grown meat substitutes, which are improving in terms of quality and affordability all the time. Hydroponics, growing plants indoors without soil, is another high-tech option, so far only achieved on a relatively small scale, but with great potential for the future. Which brings us finally to genetic engineering, the topic we're focusing on today. Okay, I began with this rather lengthy introduction because I wanted to put into context the scale of the problem the world is facing, and hence why we really can't afford to dismiss any of these solutions just because they happen to be controversial. And back in the late 1990s, nothing was more controversial than this. Dubbed Frankenstein Foods by the media, GM, genetically modified products, aroused fierce passions, mostly hostile, as the general public recoiled in horror at the scare stories which regularly made the headlines back then. And we all remember scenes like these, mass protests and direct action against the megacorporations and research establishments perceived as being in cahoots to foist potentially dangerous procedures and products on the rest of us. And this was the inevitable result. Although GM products, if properly tested, may well have been safe, the technology itself acquired such a toxic reputation that governments around the world were cowed into passing strict new laws which made GM food effectively unsaleable. And that could have been the end of the story were it not for this. A research paper published in 2012 in the journal Science with the somewhat obscure title, A Programmable Dual RNA Guided DNA Endonuclease in Adaptive Bacterial Immunity. Phew. Now, please don't ask me what this all means because I'm really not sure, but the scientific community were electrified by it. And now, 10 years on, this groundbreaking discovery has blossomed into a technology with the potential to save millions of lives all around the world. So much so, in fact, that in 2020, two scientists who led the research team, Manuel Charpentier and Jennifer Doudna, were awarded the Nobel Prize in Chemistry. And this is the technology they pioneered, CRISPR-Cas9, a procedure which utilizes so-called molecular scissors to alter the genetic code of any species, plant, animal, or even human, in a precise and controllable way. Hence the description, targeted genome editing, which, as we'll see in a moment, is a lot less of a mouthful than what the acronym stands for. Now, at this point, I was intending to explain in simple terms how it works, but after tying myself up in knots trying to summarize such complex science in terms accessible to mere mortals like ourselves, I gave up and decided to leave it to professional communicators. So here's a short video which does the job much better than I ever could.
In a document, if we suspect we've misspelled a word, we can use the find function to highlight the error and correct it or delete it. Within our DNA, that function is taken on by a system called CRISPR-Cas9. CRISPR is short for clustered regularly interspaced short palindromic repeats. CRISPR consists of two components, the Cas9 protein that can cut DNA and a guide RNA that can recognize the sequence of DNA to be edited. To use CRISPR-Cas9, Scientists first identify the sequence of the human genome that's causing a health problem. Then they create a specific guide RNA to recognize that particular stretch of A's, T's, G's, and C's in the DNA. The guide RNA is attached to the DNA cutting enzyme Cas9, and then this complex is introduced to the target cells. It locates the target letter sequence and cuts the DNA. At that point, scientists can then edit the existing genome by either modifying, deleting, or inserting new sequences. It effectively makes CRISPR-Cas9 a cut-and-paste tool for DNA editing. In the future, scientists hope to use CRISPR-Cas9 to develop critical advances in patient care, or even cure lifelong inherited diseases.
Okay, I hope you were able to follow that. This animation showed how human DNA can be edited, but exactly the same procedure can be applied to edit the genome of any other species, including, of course, plants. And with that in mind, let's now compare CRISPR gene editing with its predecessor, genetic modification, GM, which provoked the Frankenstein Food controversy of the late 1990s. GM is actually an abbreviation of GMO, which stands for genetically modified organisms, and the diagram on the left summarizes the process. A gene extracted from one species is inserted into the genome of another, and that creates what's called a chimera, which is a composite organism that could never have arisen naturally. No wonder there was such an outcry when the concept was first proposed. Gene editing, by contrast, doesn't mix species. As we saw in the film, it works like the find, cut and paste tool in a word processor. A particular sequence of DNA is located, cut out, and replaced with a new section, but crucially, the inserted DNA comes from the same species. And that's not the only significant difference between the two techniques. In GMOs, as well as mixing DNA between species, which is a completely unnatural scenario, the foreign DNA is inserted randomly at arbitrary locations and often more than once, which makes the final outcome highly unpredictable. By contrast, gene editing, if not entirely natural, the results are at least naturalistic in the sense that they could, at least in principle, have arisen by traditional breeding techniques. Finally, the specificity of CRISPR's molecular scissors ensures that the implanted DNA here is only inserted at just one precise, pre-planned location, making the whole procedure entirely controllable. And it's this precision and controllability which makes the new technology such a game changer. So now I'd like to show you a series of short videos which illustrate the transformative potential of CRISPR gene editing for global agriculture. And the first video I'm going to show really summarizes what we've covered so far and applies it to the global situation.
How can we address all these issues and still make sure that everyone and everything has enough to eat? Humans have been improving crops and livestock for centuries. Now there's a new tool, one that achieves results quickly when the climate emergency leaves us no time to spare. It's called CRISPR-Cas, or gene editing. CRISPR-Cas is a powerful instrument for positive change, a way to help ourselves and a warming planet through the wise use of science and technology. How does it work? By modeling nature's tools. Every living thing is comprised of cells. Within each cell is a genome, which contains an organism's complete set of DNA. We start with what's there, then make selective tweaks to the genome. Climate change is making it harder to grow many crops, including rice, one of the world's most important foods. So let's say we want to produce a rice variety that can tolerate erratic rainfall and drought. Using previous methods, researchers made random changes to the genome. Most of these modifications created undesirable effects. As a result, many potential plants had to be screened for their ability to tolerate drought until a beneficial change was found. Gene editing removes the guesswork. Since we know which areas of the genome may be involved in giving rice drought tolerance, we can use CRISPR to specifically edit the function of those important drought genes. Gene editing can be applied to give crops a wide range of beneficial climate-resilient traits. Crops that support sustainability and protect the environment by sparing resources and reducing applications of pesticides and fertilizer, major sources of greenhouse gas emissions. Crops that can stand up to the drought, heat, salty soil, insect pests, and plant diseases that climate change promises to bring. Crops that enhance agricultural and natural biodiversity, and crops that produce healthier versions of the foods that people know and love. CRISPR is especially important for the 3.1 billion people living in Africa and South Asia who produce the least amount of greenhouse gas emissions but suffer the most from climate change. Because CRISPR-Cas is efficient and affordable, it opens up a whole new era in agriculture, an era of equity and opportunity. One where small startups and public scientists can develop the crops that consumers want, and farmers earn the profits, not mega corporations. CRISPR-Cas offers a chance to regain more control of our food supply, improve lives, support rural livelihoods, and tread more lightly on the earth. We can produce enough for everyone and everything, even in the face of climate change, if we embrace innovation, empower local economies, and support food sovereignty.
I chose that film because I think it brings into sharp contrast the contradiction between protesters in the first world, the developed world where we live, who stand against all forms of technological advancement, and really the difference it could make to poorer people elsewhere in the world who, if the gainsayers get their way, will deprive them of a chance of a healthy life. And I think some protest groups sometimes forget that. The next film takes a closer look, if I can get it up, next film takes a closer look at one particular research project that's going on right now here in the UK.
Gene editing is a revolutionary molecular technique
that we can use to make very precise changes to an organism's genetic code.
For example, when we develop new varieties in conventional breeding, we use a lot of crosses. But when we cross this, the progeny won't have all of the traits that we want. So we need to cross again and again. So it takes up to 5 to 7 years to make a new variety. So here at the Sainsbury laboratory, we developed a project named Tomelo.
The Tomelo project is a very nice example how we can use genome editing to make beneficial changes to a plant genome. In the context of agriculture, gene editing provides a method to modify specific traits in a crop plant in a very short amount of time. And these are two very important factors, so the speed and the precision of genome editing.
So currently we use a lot of chemicals in the field to protect our crops. And the downside of this is that pathogen can evolve resistance against them, so we have less and less options. So if we can make the plant resistant, we can reduce our use of chemicals in the field.
So I was involved in making a tomato that is resistant to powdery mildew disease.
In this project, we removed a small piece of DNA from a gene that is necessary for the pathogen to cause infection.
So the way we did it is that we deleted 48 letters in the genetic code of tomato in the genes that the pathogen uses to infect the plant.
The tomato genome is approximately 1 billion letters long, and from these letters we removed 48 letters.
So this deletion made the gene not functional anymore, so the pathogen couldn't use it anymore to infect the plants. And on the other hand, because this tomato line is resistant to mildew, that means that we don't need to spray the tomato field with chemicals targeting mildew anymore.
Importantly, we sequenced the whole genome of the resulting plants, and thereby we could show that none other region of the genome was affected by the change we made. So we could really prove that we made a very specific change to the genome without affecting any other regions.
Okay, well, I found that really quite exciting. If they do succeed in producing a mildew-resistant tomato, it'll not only improve crop quality, but it'll also reduce significantly the amount of pesticides that will be needed to grow them. And the other thing I took from the film was how they're using sequencing technology to confirm that the only change that they're making to the genome is the one intended. This vital safeguard is just one of the many quality control measures that will be necessary to satisfy regulators and, equally important, to win over public confidence in such products. Right, the final film in this section, we're going to take a look ahead to some of the other potential applications of gene editing in agriculture and how the UK in particular can be at the forefront of this research. So again, just a short film.
My name's Jonathan Napier and I am a flagship leader here at Rothamsted Research, and I lead our GM program on making omega-3 fish oils in plants, but I'm also in charge of a working group that is looking at both gene editing and GM and how that can be incorporated into our future programs. So, I mean, we're really interested in trying to enhance the nutritional composition of plants. We've got several GE projects in my team looking at trying to use gene editing to improve the crop we're interested in, which is camelina, which is like an oilseed crop that's related to oilseed rape canola. And we use gene editing to try and knock out, to block a particular pathway so that the plant doesn't make something that we don't want it to make. So we're basically using gene editing as a fine-tuning mechanism to improve the oil content of our camelina plant. So what we discovered with the research we've been doing is that gene editing is really a very powerful tool to precisely change the composition of a plant. And so we used it to specifically inhibit a particular gene, and that then changed the oil profile in our camelina plants. And we did it with two different examples, one where we changed it so that the oil actually became more like you'd find in olive oil, and another example we changed it so it didn't have any erucic acid, which is sometimes found in crops like oilseed rape. So we realized that it's a really powerful technique and it's a very precise technique. I think GE has amazing potential. I think it can really revolutionize plant breeding because it can do things to a degree of precision and speed that we couldn't previously do with any other forms of plant breeding. So therefore I think it's going to really transform the way that agriculture is done, in the same way as gene editing is transforming all aspects of life sciences and the biomedical sciences. It's completely changing the way that we think about how we go about doing things. I think one real potential area that you could use gene editing for, which would be really powerful and deliver real societal benefit, would be using gene editing to remove allergens and toxic proteins that naturally occur in plants and foodstuffs. So, for example, what happened, if you could remove allergenic proteins from sesame or peanut, you could make a foodstuff that would then not be toxic, lethal to some people in society. In the same way as you could use gene editing to remove the allergenic proteins from wheat that cause celiac disease. That would be a great thing to do. Or you could use gene editing to remove ricin, which is like the second most toxic protein on the planet. you could remove ricin from castor bean. There's lots of things you could use gene editing for that would deliver real and immediate benefit to society. And I think it would be a shame not to use it.
Well, they're certainly aiming big. Enhancing the nutritional content of our food can only be a good thing, and removing allergens could literally be a lifesaver for people with intolerances. Okay, so let's pause and take stock. It's clear that the science has moved on a long way since the days of the Frankenstein Food controversy. What we need now is for governments that have so far been reluctant to embrace the genetic revolution to finally recognise the benefits and establish an appropriately balanced regulatory framework that can unlock its potential. And I'm happy to say our own government is already taking action. I said a moment ago when I started that this has suddenly become very topical. And the reason is that, hang on if I can just find my cursor, thank you. The reason is that just last month in the Queen's speech, Prince Charles, deputising for Her Majesty, announced this: "My ministers will encourage agricultural and scientific innovation at home. Legislation will unlock the potential of new technologies to promote sustainable and efficient farming and food production." And this is the legislation he was referring to, the Genetic Technology Precision Breeding Bill, which was actually presented to Parliament just a couple of weeks later. And so it's already going through the parliamentary process. And this is the bill itself. According to the blurb on the front page, its purpose, and I quote, is to make provision about the release and marketing of and risk assessments relating to precision-bred plants and animals and the marketing of food and feed produced from such plants and animals and for connected purposes. Well, that really doesn't tell us a great deal. And I have had a look. The 38 pages of closely written legalese text that follow are equally opaque. But I have managed to dig out the gist of it from the government's website. And according to that, the purpose of the bill is to remove unnecessary barriers inherited from the EU, note the pointed dig, to enable the development and marketing of precision-bred plants and animals which will drive economic growth and position the UK as the leading country in which to invest in agri-food research and innovation. Well, moving swiftly on, I wish Boris would give Brexit a miss. The main benefits of the bill would be enabling precision breeding technologies to improve the sustainability, resilience and productivity of agricultural systems. Technologies such as gene editing have the potential to increase disease resistance in crops and can reduce pesticide use, lower cost to farmers and increase food production. And the main elements of the bill, finally, are creating a new, simpler regulatory regime for precision-bred plants and animals that have genetic changes that could have arisen through traditional breeding or natural processes. And note this final clause, which I think is something we can discuss later. There is a caveat, really. No changes will be made to the regulation of animals until animal welfare is safeguarded. So that's what the government are planning. How did it go down? Well, not surprisingly, the reaction to the bill has been split down predictable lines. Agricultural institutions and the industries that fund them are all for it, whereas environmentalists and the pressure groups that support them all hate it. We clearly need some more objective viewpoints, like these. This is what the Royal Society said. Genetic technologies, including gene editing, can help address the environmental and societal changes faced by 21st century agriculture. We, the Royal Society, have always advocated that regulation of genetic technologies should be based on the outcome of any genetic changes rather than the procedure itself. So they're all for it. But for balance, I suppose we ought to hear a view from the Observer, which, as you'll know, is not usually noted for its support of Conservative governments. And this is what they said. The government's proposed legislation is a move in the right direction. It will introduce a regulatory regime for releasing gene-edited crops and animals which sensibly will now be treated in the same way as those produced by conventional crossbreeding techniques. You'll remember earlier I focused on how the products of gene editing are essentially naturalistic. They could arise by natural processes. So the Observer support, and in fact, the rest of the mainstream press all seem to support it, which is an encouraging sign. However, given the passions that were unleashed last time, the gene genie, as it were, looked like it was going to be let out of the bottle, don't expect the new bill to have an easy ride this time either. Okay, I've nearly finished, but before I do, I've got one final film to show you. It was broadcast by Sky News to coincide with the bill's launch, and it sums up very nicely all the things we've been looking at today, and it gives an advance warning, if you like, of the inevitable row that's already going to be brewing as the bill goes through Parliament.
They don't look any different to ordinary tomatoes, which is very much the point. But these have been genetically edited to produce high levels of vitamin D. Normal tomatoes, like most fruit and veg, contain virtually none. But just one of these has as much vitamin D as 2 eggs or a serving of tuna. They used a gene editing technique called CRISPR-Cas9 to tweak the tomato's DNA.
We talk about genetic tweezers, so it really is very, very small nature equivalents. It can happen naturally, or it could happen naturally, but the amount of work and time required to make that change and then to select it and breed it in is prohibitively long.
The technique is fundamentally different to previous incarnations of genetic modification, or GM. Most GM products contain genes from another organism that confer a benefit. A type of GM cotton, for example, has a gene from a bacterium to make it resistant to insect pests. By contrast, gene editing inserts molecular machinery borrowed from bacteria to delete, swap or repeat genes already in the plant's genetic code, before removing the foreign DNA afterwards. The difference is a crucial one, because this week the government will put forward a bill making it much easier to bring gene-edited crops to market. And they're closer than you might think. This plot is the first crop of gene-edited wheat growing anywhere outdoors in Europe. And the researchers behind it hope that this change in the law will be the first step to taking crops like this out of field trials and seeing them benefit society.
It's the first positive step in crop biotechnology regulation in Europe or the UK for a generation, 25 years. And I've suffered all through that period. We've been waiting for this for a long time, and I think it's really a positive step.
This wheat has been edited to make less of the cancer-causing compound acrylamide when it's fried. But researchers are also looking at editing out common wheat allergens or making the crop more drought or pest resistant.
It's really important that we don't miss out on the next biotech revolution, which is gene editing. And that is already off and running in some parts of the world. The United States, for example, several years, Canada, Brazil, Argentina. Just this year we see China and India moving on biotech, on gene editing. So really important that we keep up.
But opponents of gene editing dispute claimed similarities to traditional breeding and say the law change is a step towards more elaborate genetic engineering.
It's entirely misleading to say that gene editing doesn't involve the insertion of foreign genes. In fact, gene editing is a suite of technologies that range from a simple SNP to the complex insertion of foreign genes. And the traits that get scientists really excited, things like disease resistance and drought resistance, simply cannot be achieved without these complex technological interventions.
With gene editing, scientists have proven they've got a tool that could revolutionise food and farming, but they have some way to go to prove real benefits to society and the environment. Tom Clark, Sky News, Norwich.
So there we are. I like that report because it was well balanced. I felt, and they did give a voice to opponents. Just a word of clarification, because in that film we heard the first mention of this business about bacterial DNA. That is actually true. Cas9 is a protein that was originally derived from bacteria, and it's the tool, you'll remember from the film, which is used to cut the DNA. It is removed afterwards, but you can see the protesters, you can see how they're going to latch onto that and say, well, you are using foreign DNA and so and so and so. So expect a very hotly contested debate, but from my view, I do hope that we do see the technique becoming mainstream in the not too distant future. So that's all I've got to say. I guess now it's your turn to have your views. What do you think? So I'm going to stop sharing and hand back to Steve.
Thank you very much, Barry. Absolutely great, great talk, great presentation. I found it really, really thought-provoking, and I've got a lot of thoughts as I went through, and a lot of other people have as well. The first thing I thought of was, well, what about the meat industry? There's going to be absolutely immense objections, I think. Isn't it going to be a bit like fighting the oil industry? Huge vested interests.
Yeah, clearly. And when we cast our minds back to the GM controversy, the name that appeared most often to be sort of emblazoned across all the headlines was Monsanto. And yeah, you can understand why that company got it in the neck. For one thing, a lot of GM products, what they sold to the third world farmers was sterile seed, so you couldn't actually use these. If you grew GM wheat, you couldn't simply harvest some of the wheat and then use the seed from that to plant the next year's crop, because it was self-sterile. So you had to buy more from Monsanto, and Monsanto, of course, I had it in the neck about glyphosate, and there's a whole load of things that company. I don't want to single them out because there are others, but you can see this ogre of big business being put up and saying, well, can you trust these guys? I don't know. I hope we've learned from the Monsanto experience.
Yeah. Okay. Well, let's take a look at the chat box here. First up, John Rose, land use slide early on. It was difficult to separate dairy from beef as older cows and young bullocks are slaughtered. I don't think that's a question. It's just a comment on the slide. It's difficult to see the full picture.
Yeah, I got those slides from a website called Our World in Data, and yeah, if you go to that website, there are an awful lot of footnotes on these slides. It's very difficult to condense a lot of this information into something.
Okay, that was the next question from Brian. What website was it again?
Our World in Data.
Okay.
But they simply they collate data from reputable sources. So
okay.
There's a limit to how much one can dig back through verified all.
Okay. I noticed early on you said people don't have to worry about giving up wine. That they'll be very pleased to hear that. Stephen Birchall says, "What's the effect of producing beer?" Have you any comment on that?
I don't know. I mean, you know, of course, you think about the various ingredients of beer. You've got the barley and the hops and yeast, of course. And we know that brewers are very protective of their particular yeasts, because they claim that they give their beer a particular character that others.
Absolutely.
Yeah.
I would have thought it can only get better.
Yeah. Yeah. You could maybe give beer maybe a longer a longer shelf life. I mean, you know, sort of proper beer obviously goes off after a while, so maybe that's something you can do.
Yeah. Brian McMahon is talking about carbon dioxide in the atmosphere. It's only reported this morning it was only 0.03%. Blah blah. I'm not sure exactly what the figure is, but he's wondering if he's not questioning global warming, but is wondering if we're missing something and whether the threat is coming from something else. Perhaps the bandwagon is taking us off course. I think actually the question is taking us off course a little bit. So we'll stick with the CRISPR technology and so on. Carol says, isn't most soya that we consume genetically modified and has been for years?
Yeah. This is true. Although a lot of soya, I believe, is actually fed to animals as part of their diet supplements. Yeah, I covered the question about labelling earlier on, and the question about labelling has always been a bit of a hot potato, in the sense that if something in a foodstuff that we're going to eat is genetically modified, clearly it's important that we know about it. But if a product, say a meat product or something, comes from animals which are fed partly on genetically modified stuff, should that be on the label? I don't know the answer. These are some of the questions that the people who are legislators who are poring over this bill right now are going to have to deal with, because labelling is going to be a critical thing. People are entitled to know what they're eating, and I think we've got to make sure that we don't give the impression, I say we, governments give the impression they're trying to pull the wall down over people's eyes. I think they have to be open about this.
Yeah. Just a quick one from me while I think of it. The government legislation, the bill, the precision breeding bill that you mentioned, I mean, bullet one was all about getting the benefit out of Brexit, essentially, freeing ourselves from the constraints of the EU. Does that mean we are ahead of the curve compared to other EU countries, or are we actually pushing ahead faster than they are?
The EU introduced a blanket ban some 10 years ago. No, I don't know the exact date, but obviously while we were in the EU, and before anyone asks, I'm a Remainer, so that's my position on it. But we've left the EU, and the government is now looking for ways of kind of justifying the fall, I suppose. But yeah, now we've left the EU, we're no longer bound by EU rules, including on genetically engineered food. So yeah, we do have more freedom, but whether we'll use it wisely, we'll have to wait and see.
Yeah,
so the EU certainly are still much less, I believe they're much less accommodating at the moment about this. We're certainly the first country in Europe that's taking legislation on this through.
Okay. Jill Jenkins says, who will have control over production of these new varieties, and what safety issues will there be? Well, can you see that one?
I'm sorry. Yeah, I'm watching the chat box, and I'm also watching people waving furiously at the screen with their hands up. So Carol waving. So we'll come back in a minute. Sorry, I've forgotten the question. What was the question?
Who will have control over production of these new varieties, and what safety issues may there be? Well, Carol is waving.
Oh, Carol wants to answer. Carol wants to make a point. Go on, Carol, take it away.
Hang on a minute. Hang on. Let me.
Right. What I wanted to say was, in this country, and I'm sure throughout the EU, there are ethical bodies to which you have to submit any research proposals that use GMO and GE. And the problem now is, for the research to be approved, the problem now is that GE is a much simpler technology, much safer, and that's why the government bill is going through to enable it to be research to be done without having to go through the GMO body. I can't find out what it was called. I was looking on my phone, but there is, there is a great regulatory control on all this, existing control. And that's what the government wants to do, is to reduce the level of control on GE products, because it's unnecessary, as long as you are inserting a gene that's from the same species.
Right. Okay. Thank you very much for that, Carol. Right. Robin Clegg is commenting that the videos were from government labs. Do I detect a hint of distrust in that, I wonder? David Yule, wow, really interesting, and thank you very much for that, and more thanks below. So, oh, David Yule, while gene editing can clearly be precise, it also enabled genes from other species to be inserted. Hence, it appears the key issue in the short term is to ensure there are some limits as to which gene edits are acceptable to the public. But how can those limits be defined? Wow.
Yeah, I mean, that again, that is going to the parliamentary process. There'll presumably be a what do you call it? Those committees with expertise will call witnesses and experts to give evidence, and hopefully, and in the interest of fairness, I hope I'm sure they will listen to objectors as well as proponents of these things, and hopefully they will come up with a sensible regulatory regime that strikes the right balance. Because in the end, if government comes up with something which gives the impression of being foisted on the general public, as if there may be something being covered up, heaven forbid that this government would ever do such a thing, but in the end, you're going to have to win over public confidence. People have to buy this stuff. So, there's no mileage in trying to bulldoze it through without fully evaluating all the risks. And yes, the slippery slope argument, well, we'll start off with using genes from the same species, and then, well, why not try this one from a slightly different species, put a wheat gene into rice or something like that, and in the end you're on a slippery slope. So this has got to be nailed down very tightly in legislation. It's a bit like, I suppose, the rules that cover the marketing of new drugs. We wouldn't dream of that dangerous new drugs would get onto the market. Obviously, we've been through that over the last 50 years with thalidomide. So this is how we learn. But yeah, the regulations have got to be right. Okay, Ben.
Chris Frank has a couple of recommendations for other websites to look at if you want to see perhaps some different views from the science community. But here's one from Peter Blanchfield. He says, you might be able to answer this. When you remove a splice as a replacement for an area in the code, does it leave a gap from the area of removal?
Ooh, now we're getting a bit technical, but
yeah.
And some other people may correct me, but I believe that the way that the DNA is rejoined around the gap is actually the cell's own internal DNA repair mechanism, which it has. You may not be aware that well, you are aware that obviously that our DNA is being copied all the time by our cells. That's how our cells reproduce themselves, by copying DNA, and breaks and errors sometimes occur. And we have a very sophisticated internal DNA repair molecular technology that repairs gaps and mistakes and joins broken bits together. And I believe that is what will joins the loose ends, as it were. But you're right, once you start putting lots of other bits of stuff into the process from outside, then you do fall into this argument about, well, is all this stuff really natural? but yeah, I do believe that apart from the Cas9, which is the only foreign bit of material that's actually implanted and then it's removed afterwards, because this is all done at like a cell culture level. It's not, you're not done at an organism level. It's all cell culture, and then the cells are
okay, Barry. Yeah. I want to move on from the chat box. There's a few more down there that I'll look at, but some people are queuing up to speak. And first up is Lee. If you'd like to unmute yourself, Lee, I think you should be able to.
Yeah, okay, thank you. I've been presenting on this topic for about 6 or 7 years, so there's a huge amount I'd like to say, but I won't. One point I would like to make is all life on the planet is related to all other life. There hasn't been any evidence of any separate abiogenesis. So to talk about foreign genes is actually rather silly, because I have in my genome 40% the same genes as a banana, but I don't look yellow and rather bent. So I think but it's one of the things that the protesters latched on, and there were some very silly mistakes early days. So they took genes out of fish, put them into tomatoes to try and make them frost resistant and silly stuff like this. So there have been some blunders which the industry made, which have made things a whole lot worse. But just to make one major point, to grow food these days, it needs tons and tons of fertilizer. It takes herbicides, pesticides, and all the tractor fuel and all the time to treat those crops. Some crops need 20 applications from sowing to harvesting. My view personally is I would rather have some modified crops that don't need all that fertilizer, all that pesticides, herbicides, etc., etc. I think that as long as these foods are tested effectively, and GM products are extensively tested, far more than normal stuff, then I would rather go with that and down that route and be able to feed more people with safer food than avoid this technology. And going back to the EU thing, the EU regulations were very, very restrictive indeed, and pretty much stopped this sort of work in the UK. And that's why the government are changing the rules so that we can get some benefit from this. Anyway, I'll end up on that point.
All right. Thank you, Lee. Okay, next up, Mike Trevethick. Would you want to move? Are you there, Mike?
Yeah, oh, sorry, I've gone, my head's gone.
Oh, you've come up. You've just popped up.
Yeah, that's good. Yeah, thanks. I thoroughly enjoyed the talk. I'll just make a couple of comments first. Everything we eat is genetically modified. Every single thing that we stuff down our throats is genetically modified. Always has. Yes. It's done by crossbreeding or now genetic technology. So we shouldn't be afraid of it. I think we've got to be very careful about climate change, because we import a lot of our food into the UK. And if climate change ruins their ability to produce the crops that we import, where are we going to get our food from? That's one thing. And don't forget also is that climate change is affecting the UK. I mean, I live in the southeast, where you can see it happening every year. We go for months and months and months now and don't get any rain at all. And there was a big study which said if the UK doesn't watch it, they're going to have to move wheat production from the southeast up to the northwest or north, because we simply don't have enough rain to support the growing of wheat in the future. So someone did come around with the technology which said, you can grow wheat, but only it's going to use 10% of the water needed. That would be a massive benefit. So I think we as a society have got to get used to the fact of, if you want food, we've got to get used to these technologies, because unless you're going to get rid of 50% of all the humans on the earth, which we're not going to do, we've got to have something like this. Otherwise, we're not going to have enough food to eat. And there's so much regulation now about how you produce crops and how you test them. It's just like doing things for medicines for human diseases. There's so much regulation how you have to do it now that it's just immense, and you should have confidence in the regulation. The only thing I would worry about is reducing the regulation on the CRISPR things, because you are modifying it by what you view it, and it still should be stringent safety tests that they have to go through. But that's all. Thanks. Thank you. It's brilliant talk. I enjoyed it. Thanks.
Thank you.
Thank you very much, Mike. I'll move on to Ronnie. Ronnie James.
Thank you. One thing that concerns me is that the modern food production, for example, in the case of tomatoes, have been transferred in a lot of cases to a sort of aquaculture, which one thinks you ought to be able to produce a tomato that tastes exactly the same as one that's grown normally and organically. But in fact, they don't, because if you buy them from the supermarket, to buy the ones that Sainsbury's called round tomatoes, as distinct from square ones, they have no taste whatsoever. And in fact, to buy the large vine tomatoes, they have virtually no taste as well. So in the case of gene modification, would that in any way affect the taste of tomatoes? Or alternatively, would they be able to enhance the taste, say, of tomatoes or fruit or whatever in the same way that they can put other things in by gene editing?
Do you have any comments, Barry?
Yeah, I mean, I think that's a good point. I mean, in the end, whatever the scientists come up with, it's still got to be saleable in the sense that the public has still got to take the view, not just whether they agree with how it's made, but whether actually they like the product.
Yeah.
And if they find the product to be inferior in whatever respect, there'll always be a market. There'll always be a place for, if I can call it, naturally grown tomatoes of all sorts of varieties. So I don't think genetically modified crops is going to replace the whole of the food supply. It never can. There will always be a place for this, and certainly people are going to grow their own tomatoes. Whether they, oh, I can see Carol's waving her hand again. She wants to get in. But Steve, you're in charge of this, so I'll let you.
Yeah, go ahead, Carol. You've obviously got a response here.
Yeah. I mean, I years ago I used to work at Rothamsted. It was called Rothamsted Agricultural Station, but it still did the same sort of stuff. And anyway, the trouble with modern fruit and veg is it's picked before it's ripe, and then it's kept under artificial conditions to stop it ripening too quickly. I mean, you can have apples that were picked 2 years ago that come to market now. And, you know, they're kept under a variety of gases to stop them ripening, and then the change to get them to ripen. And that's why they taste absolute rubbish. Also, grow it yourself and pick it off the vine, or find out how restaurants manage to get such nice tasting tomatoes, because you generally, in a good restaurant, you generally do not get horrible tomatoes. Or go to Spain and just buy one off the market there. But it's to do with our food chains and how, you know, we want everything just right. And scientists, you know, so it arrives looking nice. You know, even in the packaging, in the cellophane, it's under an artificial environment to make it last longer.
Yeah.
I can't remember which gases are used. That's way back in my history. However, that's the problem. It's nothing to do with GM, and that is the problem, is that we aren't willing to go to a market, buy stuff, and recognise it might be off in 2 days' time because we bought it ripe.
Yeah. Okay, thanks, Carol. I think a lot will depend on price on the shelves. That will dictate what people buy. And if they're prepared to pay for something that tastes rather better, then fine. But if these genetically modified or edited products are to sell, then they'll have to sell at the right price. And the other thing that crosses my mind is you've only got to look at the issue of the acceptance of COVID vaccinations and the massive disinformation campaigns across social media, that's the sort of thing we can expect against this, I guess, in the longer term. But anyway, let's move on. Robin Clegg.
Thanks, Steve. Just first quick point. I mentioned government laboratories only in a positive way. The UK in the lead with frontier research at a mixture of public and private laboratories, which I'm very positive about. Rothamsted and DEFRA and so on. So I think that's just fine. But my main point is to talk about public engagement with science and technology. One of the problems and lessons learned from the GM debacle was that the public didn't get convinced, and the scientists weren't engaging well with the public, with journalists and others, lawmakers, policymakers. And this time we need to get on the front foot. We need to start having public conversations and dialogues now, ahead, not next year, not the year after, right now, today. In particular, there's a whole sort of public engagement with science and technology industry, if you like. But the main point is that there are professional communicators who are really good at helping to link the researchers and the directors with different publics, including general public, media, journalists, correspondents, and policymakers, in a way that ends up with a two-way conversation, not just here's the information, we'll convince you, and everything will be all right. No, a two-way conversation, genuine public engagement, and the sector must start now.
Thank you.
I know I used to work in this sector. I was head of public engagement at the research councils in Swindon.
I heartily agree. I think what we need is a Professor Jonathan Van Tam for gene editing, isn't it?
Yes.
There aren't enough scientists in government, for sure.
Yeah.
Anyway, John Waugh. Are you there, John?
You need to unmute, John.
John, just unmute yourself.
Is that okay?
That's it. You're on.
Good. Okay. Well, this is perhaps a fairly technical question. Several times it's been described that genes from the same species are put in gene editing. Well, my understanding is really that when additional DNA, on the occasions it is put in gene editing, it's chemically synthesized. It's just a sequence of, say, 20 or 40 bases. It's not actually whole genes from the same species. Now, this is perhaps a bit of a technical thing, but it may, if people talk about putting whole genes in, that may arouse more hackles than is necessary, because it's not putting whole genes in. It's when the cut is made in the DNA, a few bases are taken out and a few bases are put in, but it's not whole genes. It's just chemically synthesized fragments. So that, if this is made clearer, maybe that may reduce the some negative response when people talk about putting genes in. That's all I want to say. Thank you.
Yeah.
Can I just make a comment in reply to that? Yeah, it sounds like an obvious point that you're making, John, but actually, if you think about it, the process, I don't want to go into detail in too much, but each generation, the sort of sexual reproduction, if you like, entails genetic mixing of genes from the two parents. But actually, when you think about it, it's not actually mixing of genes, it is actually mixing of DNA. The genes cross over, they exchange bits of each other between them, and it's not at a whole gene level, it can be at an intergene level. So you can get naturally cross-mixing of parts of genes as well as whole genes.
I think that's...
Whether they're viable is another matter, but it can happen, so it's not...
Yes, indeed it does, and this can cause some diseases, but I think it's simply sort of statistically, you're much more likely to get crossing over between genes and genes. But I take your point, yes, it's true, yeah.
Okay, thank you very much, John. Lee, would you like to come in?
Sure, yeah, just a quick point. Again, just on the subject of foreign genes, And of course, we're focusing on food in this particular talk. But of course, there are now over 800 drugs produced using GM techniques. One of those is human insulin. And that was done by putting foreign genes, i.e. human genes, into bacteria and yeasts to produce insulin, human insulin, in culture. And then that is used by millions of diabetics all around the world. And nobody questions that. And they're injecting the stuff. And as I say, there are about 800 plus other drugs produced that way. And for some reason, the public accept that and don't get upset about it. So it's a very strange thing. When it comes to food, it can get very emotive very quickly. And I really don't understand why it is that people don't have the same feelings about drugs produced the same way.
No. Well, not many people on social media know that then, Lee, obviously. Carol.
Yeah, am I unmuted? Yeah. Yeah, just to follow on from what Lee says, it also, all these personalized drugs that you get now for cancer treatment that end in AB are monoclonal antibodies, which are all produced in genetically modified organisms, and people are clamoring to get them. So I think it's just because nobody's ever said, and so that's one thing. But what I was going to say was, I couldn't find the statistics. I was looking on my phone just now, but I think about 20% of civil servants are STEM graduates, and very few MPs have any STEM knowledge. I mean, most scientists wouldn't want to go into politics anyway, and PPE doesn't teach you very much about science, and we live in the world I live in, people don't do maths, they don't get science. There is terrible ignorance about science in this country. And I just wonder how this bill is ever going to get through Parliament when the vast majority of MPs don't understand what it's talking about. And I don't know how many civil servants there are who are going to be feeding information to them who actually know what they're talking about either. My comments.
I quite agree, Carol. It's so frustrating, isn't it, when you hear some of the things that politicians say. Absolute ignorance, and there just aren't enough scientists going into Westminster. It's nuts. That's got to improve. But Jonathan Van Tam is a hero in my book. There should be more of him. Thank you. Over to you then, Robert.
A couple of things. I saw something recently about the nutrition level in modern day food is something like 20% down on what it was in the 50s. Yeah, which is the micronutrients within the food, right, which is going to be a real problem. But getting the case across, I think, is another problem, because in my mind, it started with the climate change discussion, where everything was reduced to opinion. Yeah. So your opinion is just as good as my opinion, which is just as good as anybody else's opinion. So a professional opinion is just as valuable as a non-professional opinion, because it's just an opinion. And that, I think, is the real problem, with the climate change, with the Brexit, with the COVID virus. It's all just reduced to opinion. And controversy is what sells newspapers. Yeah?
Yeah, absolutely. Headlines.
Yeah, but what I'm saying is that what we need to do as scientists is attack this whole stupidity of opinion is just opinion, that it's your opinion versus my opinion. So you can have studied the subject for 20 years, but it's still just your opinion. Yeah.
Yeah.
I've been on the internet for 5 minutes and I found something that you never discovered. Yeah.
Yeah. Exactly. I share your frustration, Robert, as do many of us here, I know. Mary, good to see you.
Yes, hello. I've been very busy because Pitt's just come out of hospital, but I made time for you this morning because this is always so interesting. Now, you see, the thing is, we're shutting the stable door after the horse is gone. We shouldn't be trying to feed billions of people. We should be trying to reduce the number of people. We want more birth control. The American thing with the abortions, the Gaia theory that the earth will cure itself if you just leave it to it. So along came COVID, HIV, and now they're saying polio is coming back. There are just too many people. We've got to have less people. You can't keep on exponentially growing food to feed this growing number of people. We need less of us. We need to die younger. We need to have less of us. There are just too many. There are just too many.
Can you volunteer first, please, Mary?
Yeah, I'm going. No, seriously though, I mean, you know, looking at the abortion thing in America, I mean, that really is too far down the line. They should be stopping them from getting pregnant in the first place. And I think there's something basically very wrong with what parents, teachers, and all right, you can laugh if you like, but I'm quite passionate about this. There are too many people, and you've got to stop people having more people because the earth only has limited resources. I mean, there are cases going back many years of island populations of things like deer, and they have to go in and cull them because they just can't sustain the number of deer on that particular island. I'm not saying we should cull people, but we should start early on and just stop them happening. That's my view. I just wanted to say also, I noticed in your slide number 24, Barry, that the Royal Society seems to say the end justifies the means.
Yeah. Well, you jump into the carousel first then.
I'm volunteering to go anytime. I mean, but the world would miss me, wouldn't I be missed? No, seriously, I mean, I do think we've got to impress on our grandchildren, because a lot of us have them. Just limit the number of children you have. I mean, in the moment, we've all been there, those of us have had children. Oh gosh, to have children, it's a great urge and you want to do it. But you've really got to take the global view. This is a little planet. I mean, where are we going to be in 200 years' time if we just don't stop this terrible explosion of population?
Yeah. You've touched on something I was thinking of, and Tom Holloway in the chat, good morning to you, Tom. He says, looking ahead as we should, and bearing in mind there are always people who ignore regulations, wheat and tomatoes are innocent enough, but modified animals and modified humans, well, I did think at the back of my mind, but as Barry was speaking, I thought, actually, this CRISPR technology is something that could quite easily be weaponized. And I'm sure people are thinking about that at this very moment. So
yeah, can I come in on that? Can I come in, get back on that? Mary makes a very good point. She refers to what the Royal Society said. They said the regulation should be based on the outcome of genetic changes rather than the procedure itself. And that is a very good point to bring up, and it's probably why, when we looked at the legislation itself, there's this caveat that the current bill really is only concerned initially with plants, and any progress into gene editing in animals has also got to consider animal welfare factors, and that is absolutely right, and as it should be. So I think that's what the Royal Society is referring to when it gives that, when it says what it does. The means do matter when it's a pig or a sheep or whatever. It doesn't. To be fair, it doesn't really matter if it's a banana.
Yeah. Okay. Carol has her hand up again.
Yeah. So yeah,
Carol.
I go with Mary, but what you've got to recognise is that it's to do with economic development, because poor families have far more children than families that are well off, both in this country and also throughout the world. So it probably matters that we give money to other parts of the world to help, economic development, so that they might end up having smaller families. But my point why I actually put my hand up was that I have a 30-year-old daughter who is the most amazing science communicator. I'm reminding her once again to volunteer to give you a talk, possibly next year. She's very busy at the moment.
Please do.
But how? There aren't jobs out there for science communicators. I mean, she works for the Intellectual Property Office, and she's part of a team who go, well, in the sort of Easter and such. She does these Zoom meetings with graduates, final year graduates, to try and encourage them to think about the IPO and becoming a patent examiner as a career. Because she's in science, well, she was in science and technology, and 15% of them are women, and there was one Chinese bloke and one Arab, and it was supposed to be part of the diversity group, which she complained was headed up by a 40-year-old white male, which she thought was rather stupid, because she's also very outspoken as Alran. But you know, she's a very good communicator, and for years I thought that's what she ought to get into. But the reality is that you end up in an area, you know, like GE. Some people are good communicators and others aren't, and it is just sheer good fortune whether you have somebody who's a good communicator or not.
Yeah.
There isn't a career of science communicating, is what I'm saying. And I
certainly persuade her to come on and give us a talk, please, Carol.
Yeah, you know, I have just sent her again.
We'd love to have a.
My son, my oldest one, he's currently designing the lighting system for the Qatar World Cup main stadium.
Ah.
I only assume the previous contractor either couldn't do it or crashed out, because it seems the day to be doing it. He says he'll do it one day when he can think of something he wants to talk about.
Okay. Thank you, Carol. Over to you, Martin.
Thank you for that, Barry. And I expect you know, Steve, that I've always commented about how many people we have on this planet. And my understanding is when I was born, there was 3 billion. If Barry is saying when I'm 90, there will be 9 billion. That's a 200% increase in population in my lifetime. And how does the planet handle that? So the comment. The other comment was going back to how much space our food takes to grow. On one of Barry's earlier thing about lamb. Well, yes, it's fine if lamb is grown on land which cannot be used for anything else, e.g. the Welsh uplands where you don't farm, etc., etc. Although, conversely, cutting down the Amazon rainforest to produce our beef is a no-no. So anything which can help produce our food in a secure manner. And this idea of gene editing, I like the sound of as a closer to reality in what we've always done is a far better, and not as it seems there will be the monitoring around it is a good idea.
Yeah, absolutely. Okay, thank you, Martin. Over to you, John. John Moore.
I'm just going back to pick. Somebody said, "Would GE be tried on humans?" Well, the fact is, it has been done on humans. Obviously, with this, you can either do it on the germline, reproductive of future generations, or for gene therapy. But it was done in China. 2 Chinese scientists genetically edited 2 babies to make them resistant to HIV. This was illegal, and they are actually in prison. So, just as a matter of information, it has been done, although of course it's legal. And of course it's done a lot on body tissues for gene therapy. A number of gene therapy procedures now do use gene-edited cells to put into humans. So that's just, if you're interested in humans, that's a bit of information. Okay, thank you.
Okay, thank you for that info, John. Robert.
I'll just come back on the world population. The fertility rate for women in the UK at the moment is a record low. of 1.58, and I think it needs to be something like 2.1 to maintain a population.
Right.
So, you know, we do not have a population problem in this country, obviously. And I'll just throw in that in a lot of third world countries, the pension is the children. You know, that basically as you get older, your children or your grandchildren will look after you. So it's a vicious circle in those countries. And I think it's like a natural phenomenon that as living standards improve, the number of children goes down.
Yeah, I tend to agree. Okay, Mike.
Comment on the last, Robert, about our population. Our population in the UK is going up. It's approaching 60 million now.
It was up 6%, wasn't it, according to the news
so
yesterday?
it is going up. The birth rate may be going down. I guess we're getting a lot of it by people coming to live in the UK. So the population is going up, and if it continues to go up, that surely has to be a problem to providing food for us all. Because the more people you have, the more mouths you've got to feed. And as I said before, as we import most of it, climate change wrecks them, then where are we going to get our food from? And I'll take the point about reducing world population, but that's not going to happen overnight, is it? Assuming ever will. So our only way to feed ourselves, surely, is to go from some genetically engineered food. Otherwise, I can't see how we can possibly survive as a species. Sorry.
No. Thank you, Mike. Time's getting on, so we'll just take one more from Carol, and I think we'll then need to wrap it up.
It was just a comment really about what Mike just said, is that the people having all the children tend to be immigrants in the UK. And in fact, if we're not going to cull the top end, as Mary suggested we might do, we actually need immigration for people to work, to produce the taxes, to pay for our pensions, and that's just reality.
Yeah.
I'm not offering to be culled yet.
There's a lot of people concerned about population growth and so on. So please, anybody, if you think you have something to say about it and could talk about it, then please let us know. It might make an interesting topic for everybody. There's a lot of issues to debate there. Okay, well, with that, I don't have any more. Do you have anything else to add, Barry, at this point?
Can I just say that we need more ugly women?
I'm going to make a very non-PC comment here. Good-looking men tend to marry good-looking women, and bad-looking women tend to marry bad-looking men. And I'm beginning to and you tend to get more children on the ugly side.
Oh dear.
I think we've better quit while we're ahead.
Yeah, I think we're all going to get no platformed if we're not careful.
Thank you, Mary.
Anyway, people, thank you so much for coming, and thank you so much for your talk today, Barry. It was really thought-provoking, and really, you've left us with an incredible amount of food for thought, if I can put it that way. So please, everyone, thank Barry for a great morning. Thanks again.
Thank you, Barry. It was really good.
My pleasure.